Hello, and welcome to my presentation titled Using Plant and Frugivore Traits to Understand Biodiversity Across Scales and Inform Conservation. My name is Hazel Anderson, and this research was done in collaboration with Dr. Phoebe Zarnetsky, PhD candidate Beth Gernsteiner, and Patrick Bills in MSU's College of Natural Science Spatial and Community Ecology Lab. Biodiversity declines are caused by many different factors. Two major drivers are climate change and habitat loss. In order to conserve biodiversity and identify biodiversity hotspot locations, we need to know where biodiversity is highest and most threatened. Plants are essential to ecosystem function, and numerous species rely on them. So understanding their patterns of biodiversity can help identify regions for conservation that will benefit numerous other species that depend on plants. Unfortunately, we lack information on plant species conservation statuses and patterns of biodiversity. The International Union for the Conservation of Nature's Red List relies on information such as range maps and knowledge of species interactions in order to assess conservation statuses for species across different taxonomic groups. Species are assessed by putting them into different extinction risk categories, shown in the figure on the left, which include critically endangered, vulnerable, near threatened, and least concern. As of 2021, 54,127 species of plants were evaluated of the estimated 422,745 described species, which is about 12.8%. In contrast, 54,807 species of mammals have been assessed of the estimated 73,118 species, or 75%. Thus, there is a need to increase our knowledge of patterns of biodiversity in order to be able to assess more plants for conservation statuses. In particular, species in the Neotropics, especially South America, are predicted to be at high risk for extinction, as shown in this figure, and contain numerous biodiversity hotspots. The Neotropics is also home to 37% of the world's known plant species. This makes it an ideal location to focus on to fill in our knowledge gap for plants. In order to understand patterns of plant diversity, their biotic interactions can be used. Some of these interactions are very specific, and some are shared among species. The actual network of interactions can help us understand which species are more at risk of environmental change. One biotic interaction is seed dispersal, which is especially important for conservation. For example, fruiting species of plants rely heavily on animals that eat their fruits, called frugivores, for seed dispersal, and frugivores depend on these plants for food. An example interaction map between fruiting plants and frugivores is shown here for a tropical forest in Mexico. The rectangles, or nodes, at the bottom represent plants, and the nodes at the top represent birds, bats, and non-flying mammals. The width of the links and nodes correspond to the intensity of the interaction. Increasing our understanding of these interactions can in turn help us better understand patterns of biodiversity. I said that plants are important to ecosystem functioning, but what is ecosystem functioning? Ecosystem functions are chemical and biological processes that maintain an ecosystem and allow it to survive. Examples of ecosystem functions are shown in the left column of this figure and include things like nitrogen uptake by trees, water allocation to tree organs, and runoff. In contrast, ecosystem services are defined as benefits that humans derive from ecosystems. Examples of ecosystem services are shown in the right column of this figure and are benefits such as fruit production, pollination, and pest control. Most conservation decisions are made based on a simple metric, species richness. But species richness is only one dimension of diversity, and as I've explained, capturing the diversity of functions and roles that species play is also important. Therefore, we need to be looking at multiple dimensions of diversity in order to effectively quantify biodiversity. In addition to taxonomic diversity, which is often measured as species richness or the number of species, functional diversity, the number of unique traits, and phylogenetic diversity, the number of genetically distinct lineages, should also be utilized. This figure illustrates the area that would be most suitable for conservation of biodiversity and ecosystem services. There are separate maps showing priorities for taxonomic diversity, functional diversity, phylogenetic diversity, as well as a map showing priorities for all three diversity components combined. There's also another important aspect to consider when determining conservation priorities is ecosystem services. The intersection of priorities between biodiversity and ecosystem services is important to consider when making conservation decisions. Areas on these maps that are red have a value of 1, meaning they are of highest priority, and comparatively, areas colored purple have a value of 0, which are considered to be lowest priority areas. In this project, I focused primarily on taxonomic and functional diversity. Through my research project, I aim to answer the question of where are the spatial patterns of taxonomic and functional diversity most correlated between plant and frugivore species in the Neotropical country of Ecuador. I did this by combining methods from data science, ecology, and biogeography in my approach. To calculate the taxonomic diversity for plants, I used data from the Botanical Information and Ecology Network database for species in Ecuador. I then compiled occurrence maps for those species and filtered that using the forest cover from NASA satellite data to examine just forested areas of Ecuador. I also subsetted this data by species of plants with fruits. To calculate the functional diversity, I will be combining data from the BN and TRI databases for a subset of traits for species in Ecuador. The results will be used to calculate and create a functional diversity map. Parallel methods were used for frugivores by partnering PhD candidate. The figure on the left shows the species richness for all Ecuador plant species documented in the BN database. The figure on the right is an elevation map of Ecuador. There appears to be a relationship between species richness and elevation, as the highest number of species are in areas of mid-elevation, which would be around right here, which corresponds to these yellowish red values for elevation. Maps of richness in the forested area of Ecuador for plants and frugivores are shown here. Warmer colors indicate higher species richness in forested areas. There appears to be no visual correlation between the two, which is something that needs to be examined further. This could be due to the higher sampling of mammals in the Amazon, which is located on the east side of the country. These two histograms examine the distribution in richness values per map grid cell for fruiting plants and frugivores. Note that the plant richness is more evenly distributed than frugivores, but both have a higher frequency at higher richness values. Our next steps of calculating the functional diversity and testing correlations between plants and frugivores will hopefully allow us to better understand what is happening in these interactions. Future projects could build upon this research by expanding the analyses done here from our case study of Ecuador to the entire Neotropical region. Temporal data could also be used to see if the relationships and patterns of diversity have changed over time. Future patterns and relationships under climate change could also be predicted by correlating past patterns with climate data and modeling expected future changes. Here's a full list of citations used in this talk. I'd like to thank the URAF organizers for this opportunity to share my research, as well as the MSU College of Natural Science for funding, which enabled me to complete this research. I'd also like to thank my collaborators and the MSU Space Lab. If you'd like to learn more about me or the lab, visit the links below. Thank you.